consisting of 6 lectures by 6 eminent speakers of the country. We have organized in the 50th year of the Department of Plant Breeding and Genetics in Assam Agricultural University, Jorhat. On my personal behalf, on behalf of the department, as well as on behalf of the Assam Agricultural University, I convey my greetings, our greetings, as well as welcome to all the participants and the speaker. Today's speaker, as Roman has already mentioned, our own faculty, Dr. Bidyut Kumar Sarmah, is very well known throughout the country and also outside as an eminent biotechnologist now. Well, Dr. Sarmah started his career from this department. He was, after his master's in this department, joined as assistant professor. Then he went for his PhD at IIRI New Delhi in the Division of Genetics. Then after coming from IIRI, briefly he was in this department. Then he moved to the newly formed set up Department of Agricultural Biotechnology. Now he has keen interest not only in biotechnology, in the specific area, his area of interest is transgenic technology as well as the other gene technology, which I think he will highlight today. He is very much well known as for his cardinal work on development of transgenic chickpeas resistant to pod borer. Well, Dr. Sarmah is he was a brilliant student here, as well as he is also one brilliant faculty in Assam Agricultural University. Now, he is the ICR National Professor, Board of Chair Professor, as well as he is Director of the DBT-NECAB, that is Northeast Center for Agricultural Biotechnology, which was also earlier known as DBT-AAU Center. In fact, Dr. D.K. Sarmah was one of the persons who was instrumental in developing the biotechnology program in Assam Agricultural University and also building the iconic building of AAU Jorhat. Dr. Sarmah has established a number of international collaborations, and he also brought number of projects funded by international agencies like Australia, Switzerland, etc. He also had postdoctoral experiences in research in chickpea in Washington State University, then Frederick Mitchell Institute, Switzerland, then probably more importantly, still his collaborations are there, the CSIRO, Plant Industry Canberra. With this Switzerland, Australia, maybe also US institutes, he is still having collaborative works on development of resistant lines, particularly to pest, pod borer and stored grain pest in chickpea in particular and grain legumes in general. He has a number of publications in high impact factor international journals, and he has co-authored 3 book chapters. He has transferred his technology in 2009 to he developed the lines of chickpea carrying the BT genes, Cry-AC, Cry-AC, Cry-1AC, Cry-2AA, and these lines was initially transferred to Mexico in India. Then later it was also transferred to another private sector company, that is Sangro Seeds. Then the public sector institutions like ICRISEP, IIPR Kanpur, US, Harvard. Dr. Sarmah is one of the key members in the DBT biotechnology program. So I am very thankful when we proposed and requested him to just deliver one lecture in this webinar series. He readily agreed, and today we have with us Dr. D.K. Sarmah to present before you his seminar on gene technology for crop improvement. So I welcome Dr. Sarmah, and now I just request you to go ahead with your presentation.
Thank you. Thank you, sir, for elaborate introduction, in fact. And you taught me, so I still remember your classes, though you were not very much senior to us, but after your, I think, masters, you taught us the plant breeding. I still remember those classes. Thank you very much, and then thank you, Raman, also, all organizers.
Yeah, just one more trait Dr. B.K. Sarmah is having that I want to just mention at the beginning. He has not changed much in the last 40-45 years since his students' days. He is one of the person, he is very questioning. That is, he put questions in all the classes or seminars and everything, and he makes the program very lively. That is one of the characteristics of Dr. B.K. Sarmah. Dr. B.K. Sarmah, please.
I'm still having that bad habit of asking questions. Now the students, they complain. But anyway, I think I just teach them, how to interact, because that always helps. I'm sure there are a lot of students and attending my lecture. Some of you might have heard me before. So there will be repetition of slides for those who have already attended my lectures before. But I'm sure there are some new people who will, I hope they will enjoy the lecture. And then be interactive after the lecture. You can ask me any questions, whatever comes to your mind. Thank you very much. So should I share my slides now? Okay. Microphone volume is it is full now. It is full. From my side, it is full. Are you hearing me?
Other participants, please mute your microphones.
No. If it is not audible to all, then I'll stop.
No, it's audible.
It's audible.
It is audible.
Because initially it was pretty good, but now I think there are a lot of other microphones. They are on. That is why
I request all the participants to mute their microphones so that there is no noise.
Now is it okay? Should I? Okay. So have you seen my slides? The first slide is on.
No, not yet.
Not yet? But I have already shared. Yeah, I present already. Maybe slideshow. I present already. Yeah, that I did. Hang on. Yeah. Okay, just come. Yeah. Now it should come. Yes. Is it okay now? Okay. Thank you very much. So, কি কৈছা? Oh, slideshow, okay. Yep. So I chose the topic as because Purnabha sir told me to give gene technology for crop improvement. So I kept the topic like that, gene technology for crop improvement, with a special reference to chickpeas. Sir has already mentioned that we have been working with chickpea now more than 20 years to modify chickpea genome for conferring resistance against insect pest. That is why this will be a special part in my talk, but apart from that, we are going to discuss in general the gene technology for the improvement of crop plants. Uh, the questions that are going to be addressed today, whether genetically engineered chickpeas, they are protected against insect pest. Next question will be whether the genetically engineered crops are accepted globally. And we'll see what are the challenges that we, the genetic engineers, we are facing. So, the first part of my talk, the gene technology for the development of insect-resistant chickpeas. Why? Why do you need to go for, or why we selected this particular technology for insect-resistant chickpeas development? Because in chickpeas, if you see, there are two major problems. One in field, that is the pod borers, a lepidopteran kind of pest, and which causes sometimes 100% loss, yield loss in certain pockets, up to 100% yield loss. And another one is a coleopteran kind of insect called bruchids, Calosobruchus species. They damage chickpeas during storage. So you can see here, this lepidopteran pest, they lay eggs, and then the larvae, they go inside the seed. I always ask people if there are any pure vegetarian, maybe people from south or people from the north, they say they are pure vegetarian. So I ask these questions always. But if you see, these insects, they lay eggs, then larvae go inside the seed, and then adults, when they come out of the seed, then they make these holes. So, but when we cook chickpeas, we throw away these kind of seeds, where do you see the holes? But nothing is there, in fact. But the whole seed, which we do eat, there could be a lot of, insects insides, larvae insides. But I'm sorry to say that. But not to have that, we apply a lot of pesticides, during the storage conditions. And that is why in Assam it's a big problem. Why I chose this particular problem, before pod borer, in fact, we started working with storage pest resistance in chickpea. Why? Because in Assam we worship our god with chickpeas, and we offer the chickpeas in big religious gathering. But if you do not wash them properly, there could be a lot of food poisoning, and then perhaps you do get such news in newspapers very frequently. So these are the two problems, insect problems, and then till date there are no insect resistant varieties developed using the conventional breeding technologies. That is why it was an utmost need to go for, urgent need to go for development of insect resistant chickpeas using the genetic engineering. Now, to undertake a genetic engineering program, the first and foremost thing is that you have to have a genetic transformation protocol. So we use a genetic engineering, genetic transformation technology using Agrobacterium tumefaciens as a vector system. So here is in brief, the protocol is like that. You have to take an explant, and our explant is half embryonic axis with a cotyledon. And we prepare these explants, then we infect them with the Agrobacterium harboring our vectors of interest, and then we put them into regeneration and selection medium. I'm not going to go into details of the protocols. And then we look for the explants bearing the multiple shoots and the healthy shoots. Then we separate those shoots, and then here I developed a particular technique when I was in Australia actually. Instead of rooting directly in tissue culture, we used to do the grafting in vitro. You can see here this rootstock is non-transgenic, and the scion, this is a transgenic chickpea shoot. So then we establish them in pots and then transfer to the greenhouse. So this is in brief the protocol, and then this protocol is now quite reproducible, a very uh, authentic protocol you can say, and then a lot of other laboratories they are using this protocol. So my first story, because you see the protocol development started in the year 1997 when I went for my postdoc. So since then we have been still working with chickpea to modify it genetically for insect resistance. So now my first story is development of transgenic chickpeas resistant to stored grain paste. So now you see these are not chickpeas, these are the rasmas. So many of you you do love eating rasmas. Of course I am not a very good rasma lover, but I love rasma because it had given me the insect resistant gene. When I started working with chickpea, then I came to know through literature survey that one group in Australia and one group in US, they found a gene in rasma that is called alpha amylase inhibitor gene. Okay, that inhibits the amylases of insect and thereby gives resistance against the Calasobucas species or bruchids. So with that information, we contacted those people, and one of them, we are still collaborating with them, that is Dr. T.J. Higgins from CSIR Australia. So when we knew that there is gene available, then during my postdoc, we started cloning the gene for chickpea transformation, and before that, of course, it was transformed into pea, another legumes. Okay, and azuki bean, another legumes. They already transformed those legumes, and then we reconstruct the gene for chickpea transformation, and we obtain the resistance in chickpea. So this is in brief. I'm not going to go into discuss the details of molecular characterizations, but this is the gene construct we use. So those who are involved in genetic engineering, they perhaps know that this is a tDNA map, left border, right border, and this is the gene of our interest, alpha amylase inhibitor gene. And we drove this gene with a particular promoter. We expressed the gene with a particular promoter called phytohemagglutinin, which is a seed storage protein from common bean again. Okay, and why we use this particular promoter? Because this is a seed specific promoter, and we wanted to express the alpha amylase inhibitor gene in seeds of chickpea, mature seeds of chickpea, because the storage paste infests the mature seeds. And this is the terminator here, and NPT2 neomycin phosphotransferase we used as a selection marker gene, which is the kanamycin resistant genes. So once we reconstruct this gene for chickpea transformation, then we transform our chickpeas using the protocol developed, and then these are some of my transgenic lines. You can see here is the control, that is non-transgenic chickpeas, and these are transgenic lines, some of my transgenic lines, and you can see this is a southern blot, by which we confirm that some of our lines are having single copy of the gene. In fact, some had double copy of the genes, and in fact these lines were they were escaping the selection process perhaps, because we didn't get any integration in the southern blot analysis. And the same lines, when put in the western blot analysis to look for the proteins, alpha amylase inhibitor proteins, you can see this is the control. There's no protein, no gene, and then these are transgenic lines. Wherever the gene was integrated, we had a good level of expression of the alpha amylase inhibitor protein. And these two lines, where there was no integration of the gene, there's no expression of the gene. And that those who are from genetics, if there is a single copy, we expect to have a 3:1 ratio in T1 generations, because transgenic lines always will be a hemizygous line in T0 generations. That is why I'm just showing for two lines, because these lines we have taken forward. And these two lines, you can see there was single copy of transgene, and they are the transgene segregated in a 3:1 ratio in T1 generations. And this is the final slide for this particular story, that this is the control and these two are the transgenic lines, and you can see these two lines were completely protected against insect pests. I must acknowledge here Dr. Pulin Patgiri from Department of Entomology at Assam Agricultural University, who helped us optimize this particular protocol, and Sajmira Zaman, my first PhD student, I think she is hearing today. So we optimized this protocol for insect bioassay at Assam Agricultural University, and then we confirmed that my transgenic lines or our transgenic lines are completely protected against the storage pest. So after that, when we got the success in chickpea for storage pest resistance, then we got a lot of emails from different organizations. Even we got the mails and the letters from Government of India that we should take part in development of insect resistant chickpea for pod borer resistance. Pod borers, because they are the major, pod borers are the major insects in the field, and in fact more loss, economic loss is because of the pod borers. And that is why we started this program in the year 2004, that is genetic engineering of chickpea using Bt gene to confer protection against pod borers. And then our first funding came from Indo-Swiss collaboration, because DBT, Department of Biotechnology, Government of India was involved, and then Swiss Agency for Biotechnology was also involved in this particular program. And Assam Agricultural University collaborated with CSIRO plant industry, that is my postdoctoral organization where I did my postdoctoral research, CSIRO plant industry Australia. So in collaboration with CSIRO plant industry Australia, and these are the gene constructs. You can see here one beauty that what we use, because at that time there was hue and cry for using the kanamycin or antibiotic resistant gene as a selection marker gene, and that is why we use a twin binary vector. So this will be a co-transformation kind of approach. So we had two tDNA, you can see right border, left border, and in one tDNA we put the kanamycin resistant genes, and in the other tDNA, left border and right border, you can see here we put the Bt gene. We use the Bt gene because at that time we got the information that Bt gave complete protections against the lepidopteran pest in cotton as well as in corn. So those informations were available, those papers were, publications were available, and in fact they gone into the field. Bt cotton gone into the field at that time in 2004. In 2002 itself it went to the field, and that is why we started using Bt. And Bt, the cry genes are actually responsible for insect resistance. So we use the cry2AA as well as the cry1A seed. These two genes we put separately in two different constructs. And then purpose of using the twin tDNA was to eliminate the NPT2 gene during the segregation process, because we thought that these two tDNA will be incorporated or integrated into the two different positions, and then while going through the segregation process, and then kanamycin gene will be segregated out from the Bt. That was our perceptions. But let us see what we got. Anyway, these were the two gene constructs we use for our chickpea transformations to transform chickpea with the Bt genes, cry2AA and cry1AC. Now let us see the story. Here the story. Okay, so this is the PCR confirmation of cry2AA lines. I'm just telling the story for one gene because otherwise it will take a lot of time. So here are the two controls. Untransformed chickpeas, you can see it's quite clean. This is a simple PCR we wish to do. And then these are my T0 lines. So presence of the band indicator, this is the plasmid, and presence of the band in the T0 lines confirm that gene is present. But this will not confirm that gene is integrated. For that, definitely we have to do the southern blot analysis. And before southern, we should see whether there was expression, because southern is pretty expensive as well as pretty cumbersome process. That is why we looked for the expression, because we will use only the high expressing lines, which will give the complete protection against the pod borers. So you can see here, this is the negative control, and positive control we used as a Bt cotton. Bt cotton, a positive. So here is the Western blot analysis. So look for the BT protein availability. And you can see these are my transgenic lines. And there was a varied level of expressions, because some lines had very low level of expressions, some had medium level of expressions, and some of my lines had very high level of expressions. This is very much possible because gene will be integrated into different positions, and depending on the promoter, and then depending on the positional effect, the expression of the gene gets different. So that is why you can see here the variation in the expression level. And then we chose the high expressing lines to have complete protection against pod borers. And then we also look for the expression in various tissues, because pod borers, this is a field pest, and it started infecting the plants right from the vegetative stage to the even mature or immature seeds as well. Not in the dry seeds, but in all these stages, like sepals, petals, flowers, podworms. So we looked for the expression of BT in all these different tissues, and there was good level of expressions at the early stage, because we would like to control the BT and insects at the early stage itself. Now, those where we found a good level of expressions, we put them into the Southern blot analysis, and you can see here, this is my positive control here, a negative control, and these are the 6H, 6L, and then these were very high expressing line, and we got the single copy of integration. This is a double copy integration. And 6I, this is, although there was presence of the transgene, but there was no integration basically, because that was a false signals. There could be sometimes false signals in PCR. So anyway, so 6I was negative in Southern. So we just eliminate those lines. And then single copy lines, again, we look for their segregation, because, being from genetics, and we have to do all these things, all this analysis for regulation point of view. We have to show them that, you can see here 6A is a single copy line, and then it is segregating at a 3:1 ratio. Then 6L is another single copy line. It is also segregating at a 3:1 ratio. 6I, wherein there was no integration, and you can see that is why 0 positive is to 13 negative. So we looked for segregation at T1 generations, and then we advanced them. Those who are positive, we look for the homozygosity and heterozygosity in T1 generations, and those homozygous lines will make them, further showing them for T2 generations as well as T3, T4, like that, until and unless we get the completely homozygous set. So once we get the complete homozygous set, then we go for the insect bioassays. You can see this is 6A, is one of my best lines, and then these are the two parental lines as well as null lines. When we expose them to the insect, we fed them. The insects were fed with the transgenic leaves as well as non-transgenic leaves, and null, that is a null segregants. And you can see there's a lot of damage in the control as well as in the null segregants, but in the transgenic lines, there is no damage. The leaves are smiling, in fact. They are very happy. Insects could not damage them. Anyway, so when we publish these things, then, a lot of people, they showed interest. Even the private organizations showed interest to have our technology. So DBT, Department of Biotechnology, Government of India, and Assam Agricultural University, we decided to showcase our technology, and finally, the Maharashtra Hybrid Seed Company owned our technology. There was a ceremonial transfer during that time. We transferred our earlier vice chancellor, and then DBT is secretary. We transferred our technology to Maharashtra Hybrid Seed Company. It's a big barwala from Maharashtra Hybrid Seed Company. He is no more, of course. He is the winner of the two times World Food Prize winner. Okay. So anyway, we transferred our technology, but at the same time, the same day when we transferred our technology to Maharashtra Hybrid Seed Company, we transferred the technology to a public institution, that is University of Agricultural Science, Dharwad. And later on, we transferred our technology to International Crop Research Institute for Semi-Arid Tropics, ICRISAT, in 2010, then IIPR, Indian Institute of Pulse Research, in 2011, then Punjab Agricultural University in 2013. So leading organizations in country have also obtained our technology from Assam Agricultural University. So here, what I'm showing that, Maharashtra Hybrid Seed Company, they later on transferred this technology to Sangro Seeds. Of course, they are sister concern because of political reason. But anyway, so Sangro Seeds, perform. They made the introgression breeding, and then they transferred the gene into numbers of, varieties of their interest, and then using, I think, 2 varieties, they did the field trials. In 2015 itself, we completed the two field trials, and then there was 25% yield advantage. Everything was shown. But then, because of the current policies, we are stuck, of course, so we have to really think, whether we should take them further forward, or whether we should, whether the policies on genetic engineering needs to be, changed. Anyway, we can discuss these things during the part of discussion. So once we had, that was a field trial conducted by the private company, but we also, as I said, we transferred our technology to our public institutions, and Punjab Agricultural University is quite, moving forward now. So they have gone up to BC3 F2 now using our CRY1 AC line. That was the field trial we showed for CRY2A, but now for CRY1 AC also we had field trials. We are ready, almost ready to go for field trials, which has been done. The integration bridging has been done at Punjab Agricultural University. So I acknowledge the scientists from Punjab Agricultural University for this particular slide. Anyway, so as you know, from regulation point of view, that we have to confirm that there is no vector backbone in our transgenic. This is biosafety. These are all biosafety assessment has to be done. One thing is that there is no vector backbone. Second thing is that there is no nutritional difference. That is compositional analysis that we have to do. So all these things have been completed now in our laboratory, that we have confirmed that there is no vector backbone in our transgenic. This is the positive lines you can see, plasmid, and these are control, and then our transgenic lines, and there is no vector backbone. That we confirm. And another thing is that I acknowledge here the Department of Biochemistry also, because Dr. Ananta Madhav Borua, he collaborated with us for this, and then one of my PhD students, Ruby, has done these things. There is no significant difference in primary nutritional compositions, fatty acids and isoflavones. These are all recorded, and then we published this already. Yes. Yes. Sorry? You have lost my slides? I have not done anything. This is technology, okay. So should I? I hope there is. Okay. I'm ending show now. From which slide you have lost? It's okay. It is coming now, right? So okay. Is it okay now? Hello? No? What about the next slide? Is it okay? Why? Oh, Rupjiti, there is a problem. I don't know. The slides they said they have not seen my slides now.
It is now coming.
Okay. But in slideshow mode, there is a problem in slideshow mode maybe. I'm doing again. Okay. So is it okay? Okay. Let me put in food slide. Oh. Why it is so? There is some problem. Hang on. Some people can. My colleague is trying to do that. Oh, okay. Okay. Can we leave the full screen and then go ahead if it is okay for you? It is okay, right? Okay. So I'm not going to put in a full screen. There is some technological problem perhaps. So I'll go like this. Okay. So it's still blank.
Yes, it is blank.
Rupjiti, it is still blank. They say. Where is the problem? How? Why? Hang on. Have you seen now? হেৰি কৰি দিয়াচোন বন্ধ কৰি দিওঁ নেকি এইটো হা okay okay just hang on do you want us to save the changes you made? Oh, cancel? No. Okay, hang on. hang on. Uh, Again, I'm trying, present. Hello? Oh, okay. The continuity is getting lost now. That's the problem. Yeah, yeah. Okay. Have you seen now? It's coming. Okay. Let me go to my slides now. Is it okay? Okay. Okay. So I'm not going to put into slideshow now. Is that okay? If I put in slideshow, I do not know what will happen. Should I try that slideshow? No, no. Should I try to put in a slideshow?
No, it is
I'm putting
fine.
in. It it's coming now. Okay, that's good. Good. Thank God. Anyway, so this is the biosafety assessment. As I said, transgenic lines need to go through the biosafety assessment, and then we have already completed, some of our biosafety assessments here. So for the tDNA junction and then vector backbone, we have already completed the transgenic line. This is for the storage space lines. This is for the BT lines, and there was no vector backbone. This is very important information for the regulators. And then we have also done that nutritional composition analysis, and we have already published. I said, in my earlier version. Now it is a new version. I've started. Okay, anyway, I said the Department of Biochemistry, collaborated with us, Dr. Ananta Madhav, and then Ruby, my PhD student, has already published this paper in a very good journal, that is GM Food Crops. Anyway, so we didn't find any nutritional differences with the control and transgenic lines, which is very important information for the regulators. Anyway, the works are still continuing with those existing lines, and then once we get some more information from, or more advancement from Punjab Agricultural University and US Dharwad, maybe those lines will go for field test in coming years. We have also been generating now some new BT lines. Why these new BT lines? There are reasons, of course, we can discuss at the time of discussion if you want to know. But we are trying to express the BT in chloroplast instead of cytosol, because in cytosol expression we found some distortion in phenotype. Okay, some reduction in phenotype, in yield. That is why we thought there was a paper in soybean, and with that information, we tried to now express the gene in chloroplast. So there is a new construct, transformation is going on, and another construct is that the both BT genes, because maybe from regulation point of view we'll be needing a lines with double BT genes, not a single BT genes may not be approved, we never know. So that is why we have started developing the constructs wherein two BT genes together is there now, and we are transforming chickpeas for multigenic resistance. And then we have obtained some lines with expression of both BT genes at very high level. So for your information, as we have said already, that numbers of public institutions, they have from India, leading public institutions, they have expressed their interest to obtain our technology. And very recently, Bangladesh, we went to Bangladesh in 2017, and because the Bangladesh government is also expressing their interest to have our, because Bangladesh chickpea was very important, but now because of insect problem, they are not cultivating in large area. That is why we visited, along with my collaborator Dr. Higgins, we visited the Bangladesh Agricultural Research Institute, and then we have been now discussing, and there is a project funded by Australian government for this particular work. So you can see here farmers are very eagerly, they're smiling that if they get our chickpea, insect resistant chickpea sometime. Anyway, so that was the story, two stories I said, of course, for storage paste as well as for pod borer resistance, in chickpea. Now, should we say yes to the genetically engineered crop for food security? That's a big question. Why not? Because, if you see that biotech crops, now we say we officially, or, say politically, we say, genetically engineered crops, but, for better understanding, we tell them as biotech crops. We do not say genetically modified crops now. We say biotech crops or genetically engineered crops. And if you see worldwide, they are accepted now. About 190 million hectares under genetically modified crops. 18,000 million farmers, 18 million farmers, they are working with, genetically modified, food crops. And for informations, about 26 countries now, either they are growing genetically modified foods, or they are consuming genetically modified foods, or they are cultivating genetically modified foods. If you see the commercial biotech crops, 88 million hectares are under herbicide resistance. These are the main traits, either herbicide resistance or insect resistance or stack traits. Stack means, herbicide as well as insect resistance traits. So this is the graph. If you see, they are gradually increasing. Just 2017 data I am presenting. And, crop wise, if you see, cotton is in field now, sugar beets, soybean, corn, canola, papaya, alfalfa, summer squash, they are all in field now. And if you see, say, percent of genetically modified crops in the field, that in cotton, these are major crops I am showing. In cotton, 80% is genetically worldwide. And in fact, in India, more than 90% now under GM cotton. Soybean, 77% is occupied by GM soybean. Maize, 32%, and canola, 30% is occupied by GM canola. So in India, say out of the different countries, US is the first, and then the Argentina, Brazil, and then Canada, and then India's position is the 5th position with 11 million hectares of genetically modified, under the genetically modified food crops. I mean crops, that is the cotton. Only GM crop is the cotton in India, but we are still in 5th positions area wise. So what are the challenges we are facing? Because you see, few statements which are based mostly on assumptions made against GMOs, and that have led farmers devoid of economic benefit. I'm saying that, see, farmers are pretty much interested. Farmers are asking for GM chickpeas. Farmers are asking for GM brinjal, BT brinjal. But, they are devoid of economic benefit. That is why. Because, they are not getting good yield because of insect infestations. But these are the statements which are like, potential risks to human health and environment. Just an assumption. Allergy, horizontal gene transfer, to make the super weeds. What is the data? Organic is the future, and the GM seeds are pretty much costly. You say organic is the future. I would say there should be marriage between GM foods and then organic agriculture. Because, GM foods, as I said, suppose about, if you see about our work, we are going to use the less amount of insecticides, obviously. In cotton, from 8 sprays, now it is coming down to 2 sprays. Recently, the BT cowpeas are being, introduced into Africa, and the numbers of sprays are being reduced to, more than 50%. So this is why the organic agriculture should be actually, combined with genetically modified food crops. And then GM seeds are costly. Who says? If it is hybrid, obviously it will be costlier. But once we develop, once we give them to the farmers at, breeding varieties, it's a variety, then there is no question of, high cost. Initial cost is high, and for that, farmers do not have to bear those costs. Government is going to bear the cost because of funding the projects. But of course, if it is from the private institutions, organizations, they will definitely charge for that because they are investing their money. Anyway, consumer perceptions of the transgenic products need for advocacy. There has to be some social scientists involved. That is why I always say, now for any kind of, biotech, programs, there should be a social scientist always involved. An economist and sociologist should be involved so that, we do understand about the consumer perceptions. Other challenges like IPR issues, there are, for the golden rice development, Ingo Porteous has to overcome. He had to overcome 70 different IPR issues. So that's a big challenge. Development of marker-free transgenics. In chickpea, although we tried in different ways the marker-free transgenic lines, we use the Craddock system, we use the twin binary vector systems. I didn't discuss all these things in details, but it's not very easy to get the marker-free transgenics. But now, of course, there is no problem, because European Union, the greatest opponent of GM crops, they have already allowed the kanamycin resistant genes to be used. So anyway, process of deregulation is also a cumbersome process, because doing field test is not that easy. It's really cumbersome, because when our private partner did the field test, I visited those field tests, and I have seen how much care they have to take, how much cost that was involved in a particular field test. So these are some of the challenges. But question is that when we talk about health hazardness, why should you say? Because all the GM, this is the WHO's report, World Health Organization reported that all GM food crops, they have to pass through all safety assessments, and obviously they are not likely to present any risks to the human health. No science is risk free, I'm telling. So that is why there could be some risks, but we have to analyze, and we have to very specifically say if there is a date, is there any specific case where it is proved that this particular person is died for eating GM food. There's no such case. And then GM food crops in US, if you see GM food crops grown in the US, that have to be evaluated by the Food and Drug industry, drug administrations. And such measures are taken in any country growing GM food crops. So while talking about the health measures or health hazardness, I'm just showing this slide. See, while visiting Bangladesh, as you know, the BT brinjal was not allowed in India, but it is allowed in Bangladesh. And when we visited Bangladesh, I said I would love to eat BT brinjal. And yes, you see, my Australian collaborator and myself, we were eating BT brinjal. Four days we stayed there, and every day in the evening, we used to have BT brinjals. Okay. And then if you see the golden rice, see, here is the golden rice, there's a man behind golden rice, and then it will definitely solve a big problem like vitamin A deficiency. I'm not going to discuss this detail, because all of you attending this particular webinar, they all know that. But problem is that in no country still this has come to the field. In spite of requests from several Nobel laureates, see, more than 100 Nobel laureates gave a petition. They wrote to Greenpeace opposing, the GM golden rice. They wrote to them that stop opposing genetically engineered golden rice, because this will solve a big problem in the children, that is blindness. So that is why with all these things, I didn't have much time to discuss more and more examples of genetically modified food crops, but sometimes we can discuss them again. But I'd like to conclude that genetically engineered chickpeas are resistant to insect pest, which were developed at AU in collaboration with our public and private partners. And this genetically engineered chickpeas, if once it will come to the field, obviously it will bring a major change in pulse production in the country. And genetically engineered crops have been globally accepted for food and nutritional security. Of course, we have to do the regulations. Regulatory measures are essential. In fact, for many other science, there are regulations, but it depends how much we are following. But we are pretty much stringent for genetically engineered crops, of course. This is good. But we should say yes to the GE crops or biotech crops for the benefit of mankind. And with this, I'd like to acknowledge the Department of Biotechnology, New Delhi, India, then Indian Council of Agricultural Research, Government of India for funding enormously for quite long years now, for many years, for conducting our research. Indo-Swiss collaboration in biotechnology, Australia-India Strategic Research Fund, Australia, and then McKnight Foundation, the first laboratory, genetic transformation laboratory we could set up at Assam Agricultural University through the funding from McKnight Foundation. In fact, the McKnight Foundation gave me the postdoctoral fellowships, and that was the beginning. Okay, so I must acknowledge the McKnight Foundation USA, that's a private funding agencies, and of course my friend citizens, Assam Agricultural University, Jorhat, India, and all my collaborating organizations. I must acknowledge here, I must name Dr. Sumita Acharji. Sazmira is there, of course, But Sumita is still continuing with me. She was also a PhD student, and then assistant, I mean, a research scholars with me, and then assistant professor now, and then she has been leading the labs now, because I have to do a lot of administrative work, and then a lot of political things I have to deal with for getting our lines into the field. And they were of course not with me now, but they were very good chickpea transformers. One is Borsha, she is wife of one of my colleagues here, and Basanto, and then Narayana, of course she has left because of certain personnel. Yeah, but now we are training now new people for genetic transformation of chickpea. Anyway, Dr. TJ Higgins, my mentor, my guru, I must admire him always, and that he is still collaborating with us. And for our center, he is scientific advisory committee. He is the chairman of our scientific advisory committee. And then Dr. Anand Kumar, then director of NRCPB New Delhi, because NRCPB New Delhi collaborated with us for development of BT chickpea with the CRY2AA gene. That construct was given to us by Dr. Anand Kumar. And Dr. A.C. Sarmah, who did the bioassays for us, in ICRISAT. He was, at that time, he was in ICRISAT. Now he has left the Bharat Char from Maharashtra Hybrid Seed Company. And many of my students, I'm not sure whether all of them are there, but some of them are there now. Some have already left, of course. They are doing, either they got job or they are doing, postdocs in different institutions. I must, acknowledge them, their help, and their dedication. I must acknowledge. So with this, this is those who are not from AU, I must show you our center. This is initially the DBT. We are not leaving this name because, we are known as DBT Center, because DBT Northeast Center for Agriculture Biotechnology was established last year only to strengthen our original DBT AU Center, which was established way back in 2011. So with this, thank you very much to all of you. I will definitely expect numbers of questions, and I'll appreciate that. So I'm stop sharing now. Okay. Let me now stop here. Should I? Okay.
Roman, let there be direct interaction between the participants and the speaker.
Initially, the direct interactions, maybe, then I can look for the chat box. Yes, I have seen, good numbers of participations, as well as, the chat box is almost full. I don't know how many are there, but there are.
Participants would like to interact with Dr. Sarmah.
Any comment, any suggestions?
Sir, one question, sir. What is the effect of BT gene on pollinator, sir? Pollinator.
Yeah. There are a lot of papers already published. You can look for them. But for our lines, we still doing we have started the biosafety assays. These pollinations effect we have to do. I said, biosafety studies have already been initiated, and for nutritional compositions as well as vector backbone, then integrated site, those things have completed, and now we'll go for all those things. And for our lines, we cannot say.
Okay, sir. Thank you, sir. Thank you, sir.
Yeah. Manoj is asking question. Yeah. Oh, okay.
Manoj, please unmute your microphone. Dr. Manoj Sarmah, please unmute your microphone.
Yes. I'm hearing.
Sir, my
Yeah.
in Bangladesh, production is going on in Bangladesh, and BT is allowed in Bangladesh field. Now, is there any probability that BT brinjal has deficiency? We have a informal diffusion route with Bangladesh. So, is there no probability that the BT brinjal has?
See, yeah. I got your question. He said, there is, diffusible routes for Bangladesh to Assam or Bangladesh to India. We are unable to stop, say, infiltration,
Yes, sir.
very difficult, right? So, I can't say
Yes.
officially, but unofficially, I don't know.
Yes.
Unofficially, I don't know, because, when we went to Bangladesh,
But then we restrict.
yeah, when we went to Bangladesh,
Yes, sir.
I wanted to see the, BT
Yes.
brinjal cultivation as well. They took me to the areas, where BT brinjal cultivation is, at a very highest level, and those were pretty much closer to Indian borders. Pretty much closer to Indian borders. Here
Yes,
I
I
would
also
like to
saw it.
stop. Yeah. I do not want to say anything more,
Yes, sir.
because, this is official, discussion.
Yes, sir. Yes, sir.
One,
I have also been.
one Rahul
Yeah.
Saini, I have
Because
seen. Rahul Saini,
okay.
would you like to put some questions to
Some
Dr.
people said voice is not clear. Many people have said voice is not clear.
Yes.
Whether my voice was clear during my presentation?
Yes, very,
Yes, sir. Yes, sir. It was clear, sir.
Very clear. Okay, thank you. Thank you. Because some people have commented like voice is not clear. Anyway,
Rahul Saini, is there any question from you? Okay.
who is Rahul? Huh. Huh. Okay, that is IPR issues. See, cry to AA, there is no problem. I'm telling you, because we reconstructed that gene. There is no problem. And for cry1 AC also we are trying to resolve that problem, because original cry1 AC actually in India we got from Althusser, Professor Althusser. There is always a big debate on that. I have been telling about these things in many forums, but yes, we are trying to resolve that problem now, because Althusser could not claim that it was his gene, and then we have been using that gene now. So again, these are some political questions, but I cannot answer these things in details. But yes, we are trying to resolve that. Yes. See, cisgenesis is my group, as our Punobo sir mentioned at the beginning, that I am now ICR national professor. So under that, we are trying to develop cisgenic chickpeas resistant to pod borers. Cisgenesis depends on genes available within the gene pool. Genes available within the gene pool. But question comes, we are trying to now dissect the wild species as well as dissect some of the varieties reported from ICRISAT and other, organizations. That they are, say, resistant to pod borers at very low level, like 30%, 40%. Some of the wild species they have shown about 40% resistance. Now, we can definitely bring those, genes into our cultivated varieties by, genetic engineering. That is the cisgenesis. We have to use the genetic engineering technology. The cisgenics potato, cisgenics barley. People have developed these things. But question is that the 30%, 40%, resistance, entomologists will not accept them. They need at least 80 to 90%, in field level, 80 to 90% resistance. That is why we have to overexpress them. And then my group is now trying to have some high level expressing promoters from chickpea itself, because in cisgenesis we have to have the regulatory elements also from chickpea itself. That is why we have already published one paper in this direction, that we have already obtain some promoters from chickpea, and then we are trying to express these genes from wild species into chickpea cultivated species. So that is a cisgenesis. One advantage will be definitely there is no alien gene. The gene will be not alien. This is from the gene pool and from the crossable species only. So that is the advantage, maybe from regulations point of view. But in Europe also, people initially thought that that's why they moved from transgenesis to the cisgenesis. But now again they are facing same problem. Even now from the genetically genome edited plants also we are facing the same problem. In India, we are still, the regulations are not very much clear. What kind of regulations we are going to have in India for genome edited plants? for SDN1 and the SDN2, SDN3. But those who are working with the genome editing, they know all these things. So that is why it depends on regulations. As such, advantage is that BT gives 100% resistance. That is the most advantage of BT chickpea. And then we have that. But now question is, it depends on the regulators whether they will allow us to bring them to the field. Thank you. I hope it is clear.
Yes, there is one lady, Neha Sinha. Do you want to put some questions?
Introduce yourself. I request them to introduce so that I know where are they from.
Yes.
So myself, Neha Sinha, I'm studying PhD from BAU Bihar Agriculture University, Bihar.
Okay. Okay. Thank you. Please ask your question now.
I'm from food department, so I don't have any query about this tea, but I want to know just how we can incorporate this technique in food.
Into?
Fruits, sir. For minor food conservation.
Oh, okay. The only thing is that whether it is chickpea or your crops of interest, okay, your fruit of interest, you have to develop a protocol. As I showed in chickpea, initially we develop a protocol. What is the extent you are going to use? What is the technology? Whether it is agrobacterium, whether it is particle bombardment, what technology will be working for your particular fruit? You have to optimize that. And then where is the gene? That is another question. You have to have the gene of your interest, right? Once you have all these things, obviously you can optimize a protocol for genetic transformation of a fruit of your interest, and then the gene can be definitely incorporated into your fruit as well.
In fruit crops, the gestation period is too long.
Oh yes, it's long. So you take long years.
Yes,
But we will go for genetic engineering. I'm still telling if we some problems can be solved by conventional plant breeding. If the gene is available in the gene pool, go for conventional plant breeding. Or cisgenesis you go for. But if it is in your crop also, fruit crop also,
yes,
whether for breeding also, definitely you will need that long years. Whether it is conventional breeding or whether it is transgenesis or cisgenesis, the years that will be the same. Breeding cycle will be the same, right? It will not it will not vary for transgenesis, right? The breeding cycle will remain same.
Yes,
Is that clear? Okay.
Yes, sir. Thank you, sir.
Okay. Thank you. Yeah.
Dr. Sarmah, Dr. Sarmah,
Yes.
yeah, there is also a big problem in Brassica, that is
Yes.
aphids.
Yes.
You are quite aware of that. And there are some works on development of transgenic lines for against Brassica, as the gene is not available in the cultivated as well as the related wild species.
Yes. Yes.
Can you enlighten some of the
Yeah. See,
development in that line? Yeah.
Thank you. Thank you very much. In fact, I discussed with you numbers of times about this particular problem in Brassica, and in fact, I would like to go for that. But now we have seen, instead of transgenesis, whether we can go for identifying the QTLs and then go for molecular breeding. Because we can definitely go for that, but resources are very, very important. Right now, whatever the resources we have, see, my laboratory, today somebody has mentioned that apart from, I think you have mentioned that apart from chickpea, now we have also done works in other legumes. See, for black gram, the protocol is optimized, for pigeon pea, protocol is optimized, but for doing the large scale transformation work, we need big resources. We need big funds. We need people. So that's the problem. So, though I know it is important, and then genes also, we talked with, NRCP in New Delhi, as well as Boston Institute, Calcutta, that, whether they have certain, genes already available with them. They said yes. But, yes, to carry out that, definitely we need resources. But, yes, you can definitely try. I think I suggest, some people from breeding departments should take up this, particular program. I can help them. Getting the gene or getting the constructs, everything I can help. Even developing the protocol, I can help. But yes, that is one approach. But another approach is that, well, one of your students actually from your department only, Priyakshi, she is in Australia now. In fact, I helped her. We funded her initially for her PhD studies in Australia, and now they are trying to identify the genes responsible for feed resistance. And if they get the QTLs or the say genes, then they can go for molecular breeding for that. Thank you.
Yes.
Ah,
Yeah, you please go ahead.
so thank you very much from my side again. Thanks to all of you for listening to me. And I'm sure some people they are hesitant to ask questions here, but I shared my email address, and you can definitely write to me over email. Thank you
Yeah.
very much.
Thank you very much. Thank you, Dr. B.K. Sarmah. You have very nicely covered the topic, and also you have incited lot of interactions among the participants. I'm sure some other participants would also like to put some questions, but due to some sort of hesitations probably, they have not come forward. Anyway, it's very nicely you have covered the topic. Hope
Thank you.
within short period of time we will have the genetically modified food in our plate. Okay, there's maybe chickpea or BT brinjal, etc., etc. I thank all the participants from across the country for attending this webinar today on that is gene technology for crop improvement, presented by our Dr. B.K. Sarmah. Is a known biotechnologist of India as well as outside also. He is very well known. Thank you very much, everybody. Thank you.
Thank you. Thank you, sir. Thank you. Thank you, Amir.